So this is the third time I've shown you this slide. I'm showing it to you because this is really, I worry that when you're doing like Punnett squares and stuff, you're just like playing around with letters and you don't really understand what is actually happening. And at your age, you need to understand what we're actually talking about when we talk about alleles, right? The letters are referring to actual genes that are located on actual chromosomes. So when we're talking about like flower color, we have like dominant, you know, like let's say they used P, dominant, like capital P, and then like the white flower was the lowercase p. Those are referring to actual locations and actual differences in the nucleotides, the A, T, C's, and G's of those genes. And typically in genetics, whenever we're saying something is a dominant gene, we're talking about a functional enzyme, meaning it is an enzyme that will actually speed up a chemical reaction. Or in other words, it's a protein that works the way it's intended to work, whereas a recessive allele is typically referring to either the absence of an enzyme, so no enzyme, or doesn't work as well, like not as efficient of an enzyme. So the changing of that DNA sequence, that letter, would change the shape of that protein, the three-dimensional shape, so that it doesn't quite bind the substrates that it's trying to, the chemical reaction is trying to catalyze quite as strongly, so it doesn't work as well. That's what I'm trying to say. Okay. All right. Get my cursor over here. So 11.3, it's looking at, well, you know, the traits Mendel was looking at, you know, like purple and white flowers and stuff like that. Oh my gosh, I just bought a new pack of socks. I was thinking, you know, you have crop rotation in the agricultural world. Why not sock rotation? You know, so in that sink over there, I got the other socks cleaning a little bit. I'll dry them, and then that way, you know, when these socks get dirty, I'll then rotate them for the ones that are in the sink. All right, cool. Anyways, yeah, so life is not as simple as like dominant and recessive alleles. Sometimes there can be like, you can still have dominant and recessive alleles, but how they impact the phenotype, like what the actual physical trait that an organism is experiencing, can be a little more complicated. So we're going to go through some of those examples. Yeah, so an example could be like, you don't have like a truly dominant or recessive. You can have an in-between. Or maybe you could have multiple, like you could have two alleles that are both dominant. And so we'll go over that example. Or you could have a gene that has more than one allele, right? In other words, I've been telling you like for every gene you have a dominant allele and a recessive allele. But sometimes some genes could have other letters. You could have like, I don't know, like A bar, like whatever symbol you want to use, there's another allele of that gene. And what that can look like is what I was trying to say on that first slide, where maybe it's not like you have an enzyme, you have no enzyme. Maybe you have like a not so great enzyme. That could be an example. When we get into blood typing, that's a good example where you'll see the multiple alleles. Or there could be a scenario where a single gene makes multiple phenotypes. So, you know, in other words, one gene maybe doesn't just code for flower color. Maybe one gene could code for flower color and the shape of the seeds. Okay? All right. So they call this degrees of dominance, is kind of a category where they fit. Like, for example, there's complete dominance, which we have been talking about, where you have dominant, recessive. If you're heterozygous, like big A, little a, you only show the dominant phenotype. Now, there is something called the incomplete dominance, where if you're heterozygous or you're a hybrid, you're big A, little a, you will show a different phenotype, or an in-between phenotype. Okay? And then there's codominance, where you can have multiple dominant alleles. Yeah, Trinity?
So for incomplete dominance, if it was like a purple or white flower, would that be then the flower being pink? And
Exactly, yeah.
Oh yeah, and then codominance would be like, I know with purple and white, then the flower would be like a spotted purple and white?
Yeah, good. Okay. Yeah, yep. It can be a little abstract to kind of understand what exactly they mean, so you want to really, that's where I think hopefully the mastering bio, seeing some of the questions on it will help make it kind of clear. So incomplete dominance, it is like you can have basically you have more than one dominant allele. Here you have, like, notice how they're not using capital and lowercase letters, because red is just as dominant as white in certain plant species. So if you mix a red and a white flower together and you make a hybrid generation, one that has like both, they just use the color like C. I don't know why they show C, maybe for like color, but like CR and CW. So you have a red allele and a white allele. You don't show, like when we were going over like purple and white flowers, if you had like the big P and the little p, if you were a hybrid or a heterozygote, you would show the dominant, which was purple, flower trait. Here, if you're heterozygous, you show an in-between. You have a blending here. Okay. And then if you were to then cross the F1 generation, so this pink flower with itself, you would then get the reappearance of like the individual red and white flowers again. Okay. So in other words, the only way in incomplete dominance, the only way you're red is if you have two of the red alleles, and the only way you're white is if you get two of the white alleles. You have to be homozygous for those traits. You can't be heterozygous and still show the dominant trait. There is no dominant trait, essentially. All right. Then codominance. This can be a little tricky, I found, for students to differentiate the two. In codominance, it says there are more than one dominant allele. There still is more than one dominant allele in incomplete dominance. But what's happening in codominance is both alleles are expressed equally. In other words, there's no blending. There's no blending in codominance. You end up getting like spots. So like if you had a red and a white flower, and if in this plant, if the red and white were codominant, what you would get is a flower that gets red spots and white spots. In other words, at this part of the flower petal, maybe this red flower allele is taking over, whereas in this portion of the flower petal, the white flower allele is taking over. It's being expressed more strongly. Does that make sense? Okay. All right. Another example of codominance would be sickle cell anemia. This is an example that like you really want to make sure you know for the AP exam because it's very medically based. So the idea of sickle cell anemia is like you have normal red blood cells that are like this disc-like shape. And why would you want them to be a disc-like shape? As opposed to a sickled red blood cell that has this like sickle shape. What would be, just looking just mechanically at those, what would be the difference there? You know? Yeah, so think of like a drain, right? Like a pipe or something, right? Like hair gets caught in pipes. Trini, you want to add something to it?
Oh, did she say that they can carry oxygen better?
No, but yeah.
Okay.
Yeah, I mean, she was kind of saying these would get clogged more, but that would be related to the oxygen, right? Because these normal red blood cells, well, one, they just have the volume of this to just be better suited to carry oxygen. The other would be like, because they don't get clotted in the blood vessels as much, you're going to get more oxygen delivered. More red blood cells get to where they need to go. So again, think of it mechanically. You have a pipe. If you have goofy-looking things that go down your drain, they're more likely to get clogged than things that are like just a normal like spherical shape. Okay. Now, you would normally think, hey, if I have sickled, if genetically you show this sickled red blood cell, you would think that you would die off. You would not be able to survive, right? And if you're, so the S, HBS, this is referring to the sickle red blood cell. So if you're somebody who's like homozygous for the sickle red blood cell, they don't survive quite as well. They don't, meaning they don't like have as many offspring because they're going to die earlier. Whereas if you're heterozygous, because these are codominant, both of these are dominant alleles, they both express themselves, meaning if you are heterozygous where you have one normal, if you have a gene that gives you the normal red blood cell, so this is A, this is S, so HBA, HBS, if you're heterozygous, you have one copy of the normal, one copy of the sickle. What happens is you have an in-between phenotype. Like you will express both the normal red blood cell and the sickle red blood cell in your blood. But the question would be, why would there's, like, especially in like sub-Saharan Africa, you'll see a lot of individuals that have this genotype where their blood will have a mixture of normal red blood cells and sickle red blood cells. So they're at an increased risk of blood clots. So the question would be, why would that gene not slowly get removed from the population, since they're going to, like, theoretically not live as long and not have as many offspring? Is kind of the idea here. Did you?
I think there's like viruses that go around there that like the sickle cells are like something that actually virus, and that viruses like target that like the regular red blood cells.
Exactly. So malaria is will target normal red blood cells, and just by kind of freak chance, and fortunately in a way, if you have a sickled red blood cell, yes, you're at an increased risk of a blood clot, but you're then at a decreased risk of sickle. Or you have more increased risk of a blood clot, but you're at decreased risk of malaria. So in a way, they kind of wash themselves out in a sense, right? Because malaria is a huge issue in sub-Saharan Africa. So if you have normal red blood cells, you're gonna, like, your life expectancy is gonna be capped from, like, the risk of malaria. But then if you're the heterozygous, your life expectancy might be right around the same as someone with normal red blood cells. So that is why that gene keeps moving on in the population. Okay? So that's kind of like an evolutionary look at it, but understanding why this is codominance. So in codominance, blending is not the right word to use here, because each allele keeps its own genotype. You make a truly normal red blood cell and a truly sickle red blood cell if you're heterozygous. Like, really hear me here. This is not incomplete dominance, because in incomplete dominance, if you had red and white, incomplete dominance would say you got pink. The heterozygote got a completely different phenotype. They looked completely different. If you're heterozygous with sickle cell anemia, you're not a mixture. You're not like a blending of pink. You have either normal red blood cell or sickled red blood cell. Compare that to the last example where you had either red or white. You didn't have that blending together. It can be, I found like students sometimes can mix this together. It's a little tricky, but this would be codominance. Yeah?
I have a question. Do pandas have codominance, or is that just how they look?
Oh, the spots on pandas?
Yeah.
I don't actually know. Like, if you were to Google like codominance, you'll see a picture. Some people will use of the pandas and their spots. I think for our purposes, sure. I don't know if we'll talk about a little bit later, like in this like kind of lecture, that like it can be a little more complicated. Like a lot of these traits are controlled by a lot of different genes, and sometimes it isn't quite as simple as this. But sure, in the sense that you get the black and the white spots. I think that's fine for the purpose of just you thinking about codominance. Any questions here? All right. So let me make sure I've said everything here. Remember, alleles, they're just variations in the nucleotides, the A, T, C's, and G's. And this point, this is kind of an interesting one. When a dominant allele coexists with a recessive allele in a heterozygote, they don't actually interact at all. Meaning, if I go back to the sickle cell anemia example, like if you took the blood of a heterozygous individual, it's got the sickle cell gene and the normal red blood cell gene, their red blood cells aren't having a war inside their body. Like the alleles aren't like on the chromosomes like talking smack to each other. It's just that like it's the phenotype where these things come together, right? It's the phenotype where you actually see the actual red blood cell and the actual sickle red blood cell, what the gene actually codes for, where they interact. If that's a little unclear, I don't think it's a super important point. It's a little more of a technicality. So, and this last one is related to the second one. So it's that pathway from genotype to phenotype where these are going to interact with each other. It's the actual, like, in other words, it's like there's a saying, don't lose the forest for the trees. Like, don't get so focused on an individual tree that you're losing sight of the bigger picture, the bigger forest. So when you're talking about dominant recessive, you got to look on the whole organism level to kind of understand what we're talking about for some of this stuff. So related to that idea is looking at something called Taystacks disease. So Taystacks disease, it's an interesting disease. So there's an enzyme that will break down lipids in your brain. Because, you know, we talked about lipids, those are your fats, one of your biomolecules. Well, you know, you don't, if you have too many lipids in the brain, it's like having too many cooks in the kitchen. People start running into each other, efficiency goes down. In your brain, if you have these lipids accumulating in the cells, that just causes issues because then they can just start gumming up the cell machinery. So you have enzymes that will help break down some of these extra lipids that the brain doesn't need. Well, in Taystacks disease, there's a dysfunctional enzyme that happens. So if you're looking at just the organismal level, in order to have Taystacks disease, you would need 2 recessive copies of that gene. So we call you like homozygous recessive. Now, if we're talking the biochemical level, we would say if you're a heterozygous individual, like if you're big A, little a, you would technically be incompletely dominant. Because for the same idea of the like you would make this dominant would make enzyme that works, recessive you would make no enzyme. So like but like overall, the reason why this is incompletely dominant is because overall the individual would appear somewhat normal. They would have half the normal enzyme level as a person that had just 2 big A's, you know, 2 functional copies of that gene. So I'm going to say that again because it's kind of a easy thing to miss out on. If you have 2 normal copies of that gene that codes that enzyme that breaks down the lipids, you would have 100% of the enzyme. If you're heterozygous, the reason why it's incompletely dominant is a heterozygous individual doesn't make the same amount of enzyme as somebody who has 2 dominant copies of that gene. They would make half the amount. Okay? However, at like at the organismal level, they would appear normal because having half the normal enzyme amount is enough to prevent you from like actually getting that disorder and like having too many lipids in the brain. I hope that makes sense. Then the last one is the molecular level. We would say it's codominant. It's codominant because like each of these, like if you're heterozygous, each of these alleles has an equal influence on the phenotype. So the heterozygous individual, it's not as if like, again, you're normal if you're heterozygous. If this is kind of confusing, I don't think it's super important if it's kind of tripping you out a little bit. It's just something that the book technically talked about. I considered deleting the slide, but I think it's worth kind of trying to think about and getting your mind around how all these things can be true at the same time, depending on your perspective, like how exactly you're looking at it is the idea. All right. Okay. So you know that last example, the Taystacks disease. That's an example, as are most genetic disorders, where it's caused by recessive alleles. You end up getting a broken enzyme, and so it might beg the question: well, are there like any sort of genetic conditions that are caused by dominant alleles, inheriting an allele that's going to be making perhaps like too much of a certain enzyme? And there is. In fact, when it comes to like development of like a baby and stuff, there's a lot of like signaling molecules that control like where does the hand come out at, how many fingers do you have, how everything is developing. And if you have too much of those signaling molecules, that's actually a bad thing, right? So in other words, the gene, like if you had a dominant copy of that gene, you would make an enzyme that would make a signaling molecule. So if you have too much of that signaling molecule, bad things can actually happen. So an example would be babies that have, they call it polydactyly. So poly means many. Dactyly, I actually don't know what that means, but probably something with fingers and toes. Bless you. This is a condition where you have extra fingers or toes. So this is a condition where people actually inherit an extra copy of a gene that codes for that enzyme that controls like how many fingers you have. So that's an example of like how a dominant allele could actually like backfire on you. In other words, too much enzyme can be bad for you. Like it's not like you just want too much of a good thing can be bad for you. All right. Multiple alleles is the next example of non-Mendelian we want to talk about. So multiple alleles is an example of codominance. Because as I'm going through this, like if you Googled like blood typing, you'll see a slide that's like, this is an example of codominance. And you'll see other ones like, this is an example of multiple alleles. And multiple alleles is more of like a subset of codominance. Not all codominance is multiple alleles, but all multiple alleles are an example of codominance. So what's going on in blood typing, let me just jump to the slide to show you the picture of it. There is 3 alleles in blood typing. And why that's significant is most genes we talked about, you had the dominant and the recessive, right? You had 2 alleles. Blood typing, you can have 1 allele that would, they call it IA, and A will code for a carbohydrate. So I don't know if you ever thought about like what makes different blood types. What it has to do with is you have a, like, here's your circular red blood cell. You'll attach carbohydrates on the surface of the red blood cell. They're attached to the cell membrane of that red blood cell. And it's important for like your body to be able to like in cell recognition and stuff like that. So if you have you get 2 copies of the genes. If 1 copy of your gene, you make the carbohydrate A. There's another copy of the gene, another allele that can make a B carbohydrate. They're both dominant, right? That's why it's codominance. These both affect the phenotype equally, but they just look different. They have different chemical makeup. Then the recessive copy gives you no carbohydrate. So here's what can happen. If you have type A blood, there's actually 2 genotypes that give you the type A blood phenotype. You can either get 2 copies of that of the A allele, that A carbohydrate, so you would look like this, or you could get 1 copy of the A allele, so get that A carbohydrate, and 1 copy of the I allele, the lowercase I, that gives you no A carbohydrate. So either way, you're still showing blood type A because you only have 1 carbohydrate on the surface of your red blood cell. But then the same thing would be true for like type B. Okay, so it could be 2 copies of B or 1 copy and 1 lowercase I. Now this is where it's interesting because then you can be type AB blood, where if you're heterozygous, this is really why it's codominant. If you're heterozygous, each allele, whether it's A or B, equally contributes to the phenotype. In other words, the A allele stays A and the B allele stays B. Like that A carbohydrate and the B carbohydrate are each present, and they're not affected by one another. But because they're both present, if you're somebody if you have type AB blood and if you got a transfusion of blood that's type A, your body would reject type A blood. It would attack that's why you can't. That's why blood typing is important, is because your immune system only recognizes certain. They call these antigens, these carbohydrates. It only recognizes these certain carbohydrates. So if you're type B blood, you can only get type B blood, or what other type of blood? What was it? Yeah, type O. Good. Yeah, type O. Yep. You can either get only type B, or the one where you get O, they call it, where you get 2 of the recessive. In this case, this would be recessive because you're making nothing, where you make no carbohydrates. Trini?
So I guess I just want to make sure I understand multiple alleles. So it's just when there's a different variation of one of the type of genes. So with this, there's 2 types of dominant alleles, like the I.
Yeah.
Yeah, that's an I, right?
Yeah, this is a capital I. Yeah.
Yeah, there's the IA and then the IC. I mean, there's still the dominant I, but they're just kind of a little different.
Well, what would make this multiple alleles is the presence of this one, that you have a 3rd allele here. So in other words, let me go back to that codominant example with the flower. The reason why this isn't multiple alleles is because there's only 2 alleles. There's the red and the white. It's codominant because if you are heterozygous and you have the red and the white, you show both phenotypes at the same time. There's no blending here. So back to red blood cell, the blood typing, if you're in between, you're AB, you show uniquely the A and the B, so that's why it's codominant. But because you have that 3rd allele, you also could have the presence of a 4th genotype, or 4th phenotype, where you could be the O type, where you don't show any of them. Yeah?
Which one is the rarest?
Oh, that's a great question. I don't know that, actually. Anybody know which one would be rarest?
O.
O?
O is the rarest,
Yeah.
Well, technically O negative, because O positive people can get from O negative and O positive.
Yeah, that's a good point. Yeah, there's something called an RH factor, where it's positive, negative, that makes blood type even more complicated. We don't, it's not an actual objective you have to know for the AP exam, but just understanding multiple alleles. This would be an example of multiple alleles. But Yeah, it's really interesting, like blood typing, and this kind of gets into like organ, like transplants and stuff, where like you got to make sure it's the right blood type, otherwise that'll lead to like the organ getting rejected and things like that. All right, moving on to something called pleiotropy. So it's kind of a goofy sounding word, but pleiotropy is just saying you would have a single gene. In this case, it's actually looking at sickle cell anemia again. In sickle cell anemia, the gene that's going to like control those red blood cells called beta globin. And the reason why it's pleiotropy is pleiotropy says one gene can affect multiple phenotypes. So if you have sickle cell anemia, if this allele is coding for the sickle cell trait, it will affect you in multiple ways, because sickle cell anemia can lead to blindness, liver failure, and heart attack. It doesn't just impact one phenotype, right? Whereas like, you know, if you had just a gene that controlled like flower color, if that gene only affected just flower color, that would not be pleiotropy. But if it affected, you know, like flower color and seed shape, then that would be pleiotropy. Okay. Cystic fibrosis, which we'll talk about next class. That's another example of pleiotropy. Any questions on this one? All right, so this is just saying that some, if not most, traits that we have are actually controlled by multiple genes. And sometimes these genes will actually interact with each other. So like one gene can make a product, like an enzyme, that would then interact with another gene's product. That's another enzyme. Think of these protein complexes we've talked about, like the synaptonemo complex and like crossing over, the kinetic core complex. Those are controlled by a bunch of different proteins that each have individual genes, but those gene products ultimately will interact together. So you gotta in other words, life is more complicated than just kind of some of these simple Punnett squares. Yeah, or you could have a single trait that's impacted by multiple genes. And I'll show you an example of that on the next slide here. So epistasis is saying a gene at one locus alters, or let me go back to this, because I don't know if there's a separate slide on this. Like height would be a good example of this. Height, skin color. Those are examples of like multiple genes affecting those. Like, in other words, if you see like an in-between phenotype, that's like a bunch of different phenotypes. That's a good idea that multiple genes are impacting them. Okay. And that's why you can get like people with like really, really dark skin or really, really light skin or somewhere in between, because there's multiple genes. You know, multiple, like when I say multiple genes, I'm talking like multiple sets of letters. You know, you could have like 15 different kinds of letters that might just affect height. All right. Epistasis. This is an interesting one. Epistasis is saying a gene at one locus, remember a locus is the location on a chromosome, will affect the phenotype of a gene at a second locus. So the example they're giving here is like the coat color in labs. There is one gene that will control the pigment color of lab coats. So that gene, the dominant gene they're calling B, would be for black fur coat, and the recessive gene is brown. But you're like, hey, but there's also like blonde labs, so what's going on with that? Well, there is another gene that will control whether the pigment is deposited in the hair. In other words, they call this gene C. In other words, if you are C code, like capital C is color, lowercase c is no color. So let me show you the picture of it. So what's going on here is, let's say you take two black labs and you breed them together, and they switch the letters on us. They're using capital E for color now and lowercase e for no color. So here, if you were to do a dihybrid cross of these two black labs, they're heterozygous for both those traits. What happens is, you could get, if you look at this bottom right-hand corner, if you get two lowercase e's. In other words, if at that gene that controls the deposition of the pigment, that controls that the pigment gets actually sticks onto the hair, if you're homozygous recessive for the no color pigment, you will appear blonde regardless. I was about to say irregardless. Irregardless.
That's a word.
People in Texas say that all the time. Like it's all right. Regardless of if you had like two of the if you had if you're dominant for black, like 2 big B, it doesn't matter. You're not going to be like an in between color because you're recessive for that gene that controls if that black pigment will stick to the hair. It's a really kind of wild idea. Is there questions about that? So if you're at least, and then what's also interesting about this, if you're at least heterozygous for like that depositing of the pigment, you will show whatever like gene would go with like whether you should normally be black or brown. Okay, you only see it with the blonde if you get the two homozygous recessive for the E gene. All right. Okay, this slide I was kind of mentioning a couple slides ago. Polygenic inheritance. So that's genes that are controlled by traits that are controlled by multiple genes. And quantitative characters are typically associated with polygenic inheritance, meaning a quantitative characteristic is something that has a continuum. A continuum would be like there isn't like black or white. There is what? Gray. Sort of, yeah. Like there's shades of gray is what they're trying to say in quantitative variation, right? There's a continuum of these traits. So that's why like height would be an example of a quantitative character. Like height, like there isn't just tall and short. There's like taller and not so tall and me, and then skin color is another example where skin color is a quantitative character, meaning it is controlled by many genes, because you can have like different like gradations of skin color. It's not just like as simple as like black and white. I guess for skin color, gray wouldn't be the right phrasing. It'd be like brown and stuff like that.
It's a cool color.
What's a cool color?
Brown.
Brown?
Huh.
Yeah. Yeah. I'd hope so. All right, so let me show you an example of this. So here, this is not how it works. Like skin color is controlled by a lot of different genes. I don't even know how many, but it's a lot. It's not just like here they're saying let's pretend like skin color is controlled by 3 genes. They call those genes A, B, and C. And each of those genes, whether it's A, B, or C, they're just saying they make they just make pigment. So like A will make some pigment, the B gene will make some pigment, and the C gene will make some pigment. And if you're capital A, capital B, or capital C, that would equal pigment. And if you are lowercase a, lowercase b, if you're recessive, you have no pigment. Right? And so this is polygenic, meaning if you were to look at like the sperm or the eggs, what could happen is, or this is looking at an individual, this individual, if they're heterozygous for each of the 3 genes, what that means is they only have 3 out of the 6 possible pigments made. Right? Because think about it. Like this lowercase, this capital A would give you that pigment. This lowercase a is going to produce no pigment. Capital B makes pigment. Lowercase b makes no pigment, and so forth. So in other words, if you're an individual that was like dominant for all of them, you would have 6 black circles there because you'd make all the pigment possible. All right, so then if they did a trihybrid cross, which is just kind of bananas, they did something like that, you could end up getting all these gradations of skin color. And as I said, skin color is controlled by many, many different genes. Let's say it's 15 genes. You would have I don't even know what a 15-something cross would be. You'd have a gigantic Punnett square, and that's why skin color is so complicated, right? You see all kind of the different like continuums of from like black to white when it comes to skin color. Questions about this? Almost done here. A couple more slides. Oh, last thing would be how do genes and environment interact? If you take like a psychology class, this is the big thing to talk about. Like, is it genes or is it environment? Like, if what makes you, like, is it the genetics you inherit or is it how the environment impacts you? And you know, it's a little bit of both. Some traits are more influenced by your genetics. Others are more influenced by your environment. A good example is like height. If you go to like a third world country, or even if you look at data on height, like back in like the 1800s or something, people were a lot shorter then. You know, they're like my height versus now. And why is that? Why were the people? Did they just have different genes back then in the 1800s that like got like evolved out of the population compared to now? Why are we like taller now on average than back then? A bit to you?
It's mostly like access to like nutrition, like farming has like improved a lot, so there's more accessibility, and then like less people get taller.
Exactly. Our environment is better now to maximize our genotypes. If we can get the most out of our height genes so that we grow to our maximum height. Whereas a lot of people, not even just the 1800s, but just like kind of throughout human history, haven't been able to get like the best nutrition possible to maximize their height. Okay. So there you go. I think it's not a super complicated idea, but it is a very interesting one to think about. You know, especially like you think about things like you know, like there's a lot of like mental illnesses that could be there is the literal genetic component. They can like identify people who like maybe are alcoholics or maybe suffer from bipolar. They can identify specific genes that like influence that, but then the environment, like maybe if they grow up in a super good environment, they don't always express that that trait, but then something can kick in that would cause that to happen. Like speaking like personally, like one of my family members has like bipolar, and it's not something like we knew really at all. Like it's not something that she really expressed that much of like throughout life, but then an environmental thing, like she like ended up going through a lot of like kind of just some difficult life circumstances where all of a sudden like maybe like those pre-existing genetic conditions kicked in, where then she started expressing that bipolar disorder, where like actual like kind of brain chemistry started changing, and she started showing a lot of the symptoms of bipolar. So it's very controversial is not the right word. It's a very difficult thing to completely parse through and understand, especially when it comes to like treatment of things like that. Yeah, so whenever if you have a trait or like a character that's controlled by genetic and environmental factors, we call that multifactorial. There are multiple factors, genes and environments, multiple genes, multiple environmental factors that can control, are you going to express that phenotype? I think like the positive part of this you could think about is like your genes don't like ultimately like doom you to like whatever, like if you have like say they ran a genetic like screening and they're like you have a what's the word I'm looking for? You're predisposed to like depression or like bipolar or alcoholism. Like they've shown a genetic correlation of that. That doesn't mean that you're like doomed to become that, right? There are environmental conditions, like the things that are under your control that you can do to help like not become like what your genes might suggest you could become, if that makes sense. Okay. I think that's really about it. Yeah. Alrighty.